any change to genetic information constitutes new information.
Well, then we'd have to discuss what is new information, at least what it looks like, or what it would look like.
Yeah, information is purposeful and functional. It causes purposeful and functional things.
So you can't have a duplicated gene that is then modified to have a new function? Is that impossible?
I don't think anybody has observed such thing as a new function arise by a mutation.
What about a modified function?
That's modification of existing information, and it's typically degradatory.
Well, that's primarily how evolution works, by descent with modification.
What's the evidence of that? Scientific evidence for that?
For mutations?
That mutation causes structural design change over time in creatures. If it's not a philosophy, if it's not just your philosophy and it's scientific, then what's the scientific evidence for it?
Okay, so just how it is possible to get, for example, different varieties of brassica, broccoli, kale, and all that from a wild mustard by means of descent with modification. Yeah.
Okay, so there's morphological differences between plants. This has been known for a long time. What's anatomically different about the broccoli?
But that's a change in anatomy. It's the same thing.
How is it changed in anatomy? What new anatomical feature arises in the broccoli?
You have the in the broccoli. You have the flowers are clustered together to form like a bulb. So that's a new.
Okay, so it has a differently shaped bulb, but it's still a bulb. The same tissues in the same place. They are pressed together in a different shape. They develop as a different shape. It's the same tissues in the same location of the same body plan. What's the evidence for a new anatomical feature?
Same location, though.
Well, sure. It's at the end of the stems. It's not on. It's not in the root system. That's the same location of the body plan of the plant.
You have tulips that have the modified root system.
Yeah, and there's all kinds of evidence for changes in cauliflower too. Cauliflower does the same thing broccoli can do. It can grow in some pretty weird looking shapes. But where's the evidence of anatomical difference? There's no anatomical feature arising there in the broccoli or in the cauliflower.
Drawing that line arbitrarily.
No, I'm not. It's claimed that evolutionists often claim that changes to shape constitutes an anatomical change, but it does not.
Yeah, I'm not understanding the question really, because as I was describing, evolution is descent with modification. So every new trait is just a modification of an existing trait.
So that's every philosophy, but then you need to back it up with science and show that mechanism actually happens.
I just did. I just showed you the example.
I explained to you that's morphological, not anatomical.
Okay, so if you want to take it on a macro scale, for example, to get from a hand to a wing, you just need to modify the hand. It's the same thing.
You're welcome to believe that, but what's the scientific evidence for that?
I mean, well, you have, for example, you have the transition from theropods to birds that shows the.
Can you explain those, please? Can you name one?
You want to name a transitional form?
A transitional form to birds.
Okay, so Archaeopteryx would be the primary transitional form.
Evolutionists themselves now acknowledge Archaeopteryx is not a transitional form to birds. Try again. Try again.
Who said that?
No, I can tell you what they actually say about it.
Yeah, they do say that. I'll provide you the evidence in just a moment.
I would love.
No, they say it's likely not a direct ancestor, but it represents the type of form that you would see.
Right, so
the transition, and that's how they found it.
Right. So they're admitting it's not a transitional form. You just stated it. There you go.
No.
Transitional form.
Yeah.
No, you're twisting the words.
And emotion go at it, so it's one-on-one.
No, you just acknowledged, Taylor, that they don't believe that it's a transitional form in the evolution of birds anymore.
That's wrong.
No, you just admitted it.
Regardless of what lineage it's in.
No, you just admitted that it's not a part of the transition
No.
from theropod dinosaurs to birds.
Yeah, but that doesn't mean it's not a transitional fossil.
Well, you're welcome to believe that, but evolutionist scientists no longer state that Archaeopteryx is a transition of birds.
No,
Yeah, because evolution branches. Sorry, go ahead.
Regardless, they acknowledge that example has been refuted.
No, a transitional form need not be the direct ancestor to its descendant form. It's what it represents then. It doesn't have to be the direct ancestor. Like Tiktaalik, for example, is not the direct ancestor of modern tetrapods, but it's a transitional form.
Okay, I didn't hear what you said. I'm sorry.
Oh, sorry. Yeah, I have a bit of an accent. I'm from the Caribbean. I was saying that a transitional form is not the same as an ancestral form. Transitional form is to show a change in features, to show that a change in features has occurred.
Okay, so you're welcome to believe it's an ancestral form, but it's not considered a transitional form anymore.
No, I'm not saying it's an ancestral form. I'm saying it is a transitional form. It is not. Archaeopteryx is definitely not the direct ancestors of birds, because there were birds before.
That's the point. Then it's not a transitional form to birds.
No, it's still a transitional form.
You're welcome to believe that, but the evolutionists acknowledge that it's not a transitional form to birds. Okay.
No, they you just said that they said that
yes, they do.
They acknowledge it's from dinosaurs.
They acknowledge, they might believe that, you know.
But definitely not ancestral.
But it has bird features, and that's all it has. You're welcome to believe it's
dinosaur
something
with bird features. Right.
You're welcome to believe that. Okay. So I mean, if we examine the fossil record and all you've got is Archaeopteryx, and that supports your whole evolution theory, it's failed.
No, I mean
it's a bird.
Into the forms now, I can't name it for you.
Even the world's foremost expert on birds, Alan Fiducia, acknowledges Archaeopteryx is no transitional form at all.
Right. So
we
is the minority. The consensus is that it is a transitional form. It was always
good luck trying to show that it's a transitional form. They believe it's a transitional form simply because it has what is believed to be a spiny tail and teeth. But there are birds with teeth today. All the other features of the animal is strictly bird. It has bird features. It has bird feet. It has a curved breastbone. It has hollow bones. It has a beak. It has feathers. And the difference is it has teeth. There are birds that live on this planet right now that have teeth. So you can't say that's okay.
There are no
and it has a spiny,
predicted by evolution.
and it has a spiny tail. So
there are no extant birds now with teeth. No.
There are plenty of unique creatures that God made. That doesn't show that they're transitional. This creature might have been unique in that it had a spiny tail, spiny tail birds are now extinct. But that doesn't by itself evidence evolution.
Okay, so now by itself, in isolation, it would not be evidence for evolution. But remember, it was predicted by the theory.
Good, thank you. I'm glad you acknowledge that.
If it would be true that, for example, birds descended from reptilian forms, at least that's what Darwin said, but now we know it's dinosaurs. But if it descended from reptilian forms, then we should find birds with more reptilian-like features. So like the teeth and the long tail and the lack of the beak, and instead of the they don't have a beak, they have like a reptilian.
If evolution were true, there should be a plethora of transitional forms between theropod dinosaurs and birds.
Right, and that's the same thing Snake was trying to explain to you.
There aren't any.
There are.
The reason why we don't see
show them.
All right. Go ahead. I'm pulling it up.
Okay, so the journal Nature has published the Esionopteryx, however, is not the first discovery that shed status on Archaeopteryx. 2011, the journal Nature published a discovery of another chicken-sized dinosaur named Zyotungia, and had earlier had earlier undetermined the first bird status of Archaeopteryx. In other words, Archaeopteryx is not a first bird.
Correct.
It is fully a bird, okay? And it's
Right.
not considered the first bird. So it's not transitional form to birds. It is a bird, okay?
Okay. So
Not a
you are
transition.
correct. Archaeopteryx is definitely not the first bird, and is definitely not the direct ancestor to birds. No one is saying that.
Excellent.
That's not the meaning of a transitional form.
It is a bird.
Form is a species or a taxa or whatever that would show the transition from one state to another, from one kind or whatever.
Well, you're welcome to believe that, but evolutionists acknowledge it's not transitional to birds.
No, it's not ancestral. It's not.
They say it's not transitional to birds either.
It is a transitional form. It's just not ancestral.
Well,
It's
you're
not
welcome to believe that, but all I'm pointing out is they say it's not a transitional form to birds. So it doesn't help the evolutionist case that there's transitional forms to birds in the fossil record. You have to start with something else. You can't use Archaeopteryx. The evolutionists admit it's not. It's fully bird.
Who said that? Who said that?
I
Fiducia?
just quoted you a paper in the journal Nature.
Archaeopteryx. That is true.
Okay,
Archaeopteryx.
the British Museum of Natural History in 1979 acknowledged it. Okay, so Alan Fiducia, the world's foremost ornithopterist, whatever the name bird expert is, I've forgotten, ornithologist, he says the same thing. Let me find you some more statements. And
is this a consensus, by the way, or is just is this a consensus now? The current
Pascal Godefort of the Royal Belgian Institute of Natural Science agrees. I'm just looking at some of my sources. Okay. So
But you're missing the point, Neff, which is that
no,
since
you're
they
missing the
occupy
point.
these transitions, can I finish? Since they occupy these transitions where they have features of both, that shows that the phenotypic plasticity overlaps between all of these creatures.
Okay, so you're welcome to believe that there are transitions, but Archaeopteryx is not one.
What did I just say?
You're trying to use phenotypic plasticity as a mechanism for evolution, but you never answered my question. You said genetic mutation does it also. And so I asked you, what is the scientific evidence that genetic mutation designs new structural designs in the anatomy of living things? You have not answered that question. Are you going to answer it, or are you going to tell me that you don't know the scientific evidence that verifies that?
Because mutations cause differences in the regulation of the genetic material.
That's true. Mutations
in the instruction of the genetic material.
That can happen. So now, what's the scientific evidence that mutations cause changes to anatomy?
I find it strange that I've given you an example of this, and it's not
you
enough.
gave me Archaeopteryx.
Because
it's not an example
hold
of
on,
a transitional form to bird.
Because the anatomy genes can change. Do you understand this?
Oh, they can change, right? So now, what's the scientific evidence that mutation changes anatomy?
So you're asking for like a specific one.
See, we know that mutations can change genes. You're not giving me the evidence that mutations cause structural evolutionary change.
I'm giving you the mechanism.
Well, that's what you believe is the mechanism. Now we want scientific verification of your belief.
Yeah, we know mutations happen.
No, you know in your imagination. We know in scientific evidence, Taylor. If evolution is not a philosophy as I state, and so many creationists do, and it's actually science, then don't give me a philosophical idea and call it science.
So mutations do not happen in anatomical genes? Is that what you're saying?
Mutations do occur in all kinds of genes. Now, what's the evidence that mutations occurring to genes create new anatomical features? I understand you believe that. But what you believe and what's actually true might be two different things.
Because once you have a muscle gene, you can place a muscle, or once you have a bone, you can place multiple bones in different locations. And all you have to do is tweak that information a little bit.
I can build all kinds of things with my Lego set. Doesn't mean they arise naturally that way.
No, Legos don't. They don't reproduce, and they don't reproduce with variation,
Right.
and they're not
Right.
subject to principles of descended modification.
Right. So Legos don't even
they've
come
written
close
programs.
to the ingenious design of living things. I agree.
But listen to this. They've written programs where Legos do build themselves and they reproduce inside the program and they actually evolve new forms.
No, what they did is they wrote an algorithm in there that's capable of assembling them in different ways so that you get different shapes that rise.
Right.
That
Intelligent origin
doesn't
is
have any
different from
bearing on
evolution.
what we observe to happen in living systems with genes. What's the scientific evidence that mutation builds anatomical features? You see, the
Just
simple
repeat the
answer
Lego
is
example.
you can't answer the question, so I'll answer it for you. There isn't any. That's just what you believe. That's your philosophy of evolution. That's not scientifically verified. That's your belief. I understand. Now, so I have some questions for you.
Produce results in science. You don't.
Well, I mean, there's no results that show mutations build new anatomical features. That's for sure. There's no scientific results for that.
We've been over them.
Well, you haven't mentioned one. I've been asking for an hour. How many hours will it be before you can give me one?
The dog eyebrow muscles?
You don't know of one. No, I told you. You acknowledged that wolves already have these genes and produce and have these muscles.
Right. They have
Right.
muscle genes, so
Okay.
they can place a muscle in a new location.
Well, how do you know there's a new location? No, you acknowledge some wolves already have these.
And some don't.
And some don't. Okay, so phenotypic plasticity might be the explanation for that. That doesn't mean that it's been demonstrated that genetic information arose that created a new muscle in a dog's eyelid.
It's just a tweak of the already
So you say.
muscle gene that it already has.
So you. Oh, thank you. It's already got the genetic information. Next.
Yes. Yep.
Phenotypic plasticity is not evolution.
Call it whatever you want.
Process now.
All right, I'll be right back.
Phenotypic plasticity is not evolution.
But it's part of evolution.
I understand you believe that, but you can't show that with science.
What do you mean? Okay, so when you said explain how these things happen, you want me to explain the exact mutations as how these things happen?
Well, I mean, a controlled example or something, but something scientific. I mean, you're claiming this is scientific. You're claiming science verifies these things, but we don't get verification of that from you guys. We just get you guys telling us what you believe, and then you're declaring it science, and then you're calling us ignorant for not believing it. We don't have any evidence for the things you say. These are your ideas in your head. That's why evolution is a philosophy. It's not science. See, it's none of it's observable, testable, and repeatable. You can't observe, test, or repeat evolution theory in any way.
Okay, so all the variation we see within the barons or kinds, I'm not sure what you call them, is that all down to plasticity? Does mutation have any effect on the thing?
Plasticity is an organism has genetic information. It can express it more or express it less. That's not a mechanism for the creation of new genetic information that specifies a new structural design in the anatomy. That's Are you trying to misrepresent phenotypic plasticity? Are you claiming that phenotypic plasticity goes all the way down to the point where it includes the rise of entire sets of genes that code for a new structural design? Is that what you're claiming? That phenotypic plasticity includes the origination of a new anatomical feature by new genetic information? Is that what you're claiming?
No, I'm actually asking you, what do you think that the changes you see within the Barrowmans is a result of? Is it a result of pure plasticity or expression?
Well, the changes that occur to living things are variation produced by variable expression of the organism's genome. And phenotypic plasticity can mean that a trait either appears or disappears in an organism because of its ability to variously express the information that exists already in its genome. Right?
Okay.
So now that information for that feature to arise in the organism's anatomy,
right.
it must have the information to make it. Now, evolutionists are not able to provide a mechanism that creates that genetic information.
The genes come first in the expression. But
that's correct.
Right. So that's how evolution works, actually. The genes come first and then the expression.
So what's the evidence that genetic mutations build new information that codes for new anatomical features?
Right. So you want something that codes for a new anatomical feature.
Well, that's what you're claiming happens. I'm not the one who makes that claim. See, the simplicity of this is that you can't provide a scientific evidence for what you're saying. I understand you guys believe that. You believe a philosophy, a chain of thinking. You believe a way of thinking about things. What you don't, what you're not, what the thing is, there's just not any scientific support for it. It's what you believe. I understand. It's a package of ideas. You will think, well, this makes that happen. And, well, this obviously is why that happens. Well, those are your ideas. But that's not science.
What about
that's philosophy?
I'm trying to get around your argument because the thing is, I've shown you new anatomical features, but you
did? Where?
Them as anatomical features.
Well, I don't remember you showing me any new anatomical feature. What are you talking about?
A tulip.
Tulip.
A broccoli, a cauliflower, and so on.
No, that's not anatomical. That's morphological.
What about baleen? Whale baleen?
Whale baleen is an anatomical feature.
Is that an anatomical feature?
Yes, it is.
That's due to a mutation in the tooth gene.
It's expressed by genetic information during fetal development. There's sets of genes that make it. So where's the evidence that the genes that make the baleen in the whale's mouth arose by random mutation?
Because it's a piece of the tooth gene that creates these basically strands of tooth.
So it uses some of the same genetic information as what's used to make teeth.
Yes.
Okay, so what's the evidence that that information arose when it didn't exist before?
Are you talking about the original teeth?
No, I mean, there's a sequence of genetic information in the human genome that creates 576 different proteins from one string of genetic information. Nobody argues that's because of overlapping and compressed information. Why should I think a whale's genetic genome is not compressed as well? You're not providing an argument here.
But that's precisely how evolution works.
No, what happens in your imagination is that whales didn't have baleen once.
Right, so what I'm trying to say is that traits are modified. They're not added. That's how you think they're added. They're always modified. Every single trait in a species is just...
Okay, so let's examine that idea. So if that's true for species Z, then species Y, had its anatomical features because it had the same genetic information. Let's start the other way. Let's start with species A. Species A has X amount of genetic information. It has X genes, right? Now, species B comes into existence with new anatomical features by recombining the genetic information that was in species A. Now, species C comes into existence, and it has also different genetic anatomical features, and it came into existence by recombining the information that was in species A. And species M comes into existence by recombining the genetic information that existed in species A. What you're positing is that the first creature had all the genetic information for all anatomical features of all living systems.
No.
That can't be true. If evolution were true, it would have to give rise to new genetic information continuously.
Right. Evolution can... I'm not too sure what I mean by new information, but talking about new genes and so on.
So,
and the expansion of the genome, yes. Not necessarily, because, again, organism complexity is not necessarily... is not directly proportional to genome size.
So what I'm pointing out is that your argument that recombination is what does it is insufficient.
Well, not just
You still have to. If
recombination,
evolution were true, I understand that recombination would play a role if it were true. But that's nonetheless, evolution does still require that new genetic information arises, because recombining the existing genome of an organism is not going to give you the new information that makes new anatomical features. You're not going to get lungs in ichthyostega by recombining the information in Eusthenopteron's genome. You're not going to get a land amphibian tetrapods lungs by recombining the genetic information that makes gills and Eusthenopteron or Pandorixis. Okay? You understand me?
So Eusthenopteron and so on, and all these kinds of fish, they have a kind of float bladder that acts like a lung. So they actually had some proto-lung, to say.
Okay.
And that was then modified into lung. So it's just a modified version of something else.
Well, swim bladders exist. Even coelacanths have swim bladders. Okay, but you're telling me that recombining the genetic information that makes a swim bladder will create a lung.
Right. If you modify.
So what evidence is there in science that this kind of recombination creates new anatomical features?
I just gave you an example.
No, you gave me two creatures that exist in the fossil record, which are dramatically different. One is ichthyostega, and the other is Eusthenopteron. What I need is a scientific explanation of how we have verified that recombining the information in Eusthenopteron was going to create the lung in ichthyostega. Show with a modern example. Can you do that?
Yeah, we have several species that can absorb oxygen through their esophageal tissues, which is connected to the stomach. Sorry, go ahead. I just wanted
Great.
to
So creatures are designed to absorb oxygen. Interesting. So there is no scientific verification of your idea that recombination of genetic information will create a new anatomical feature. What's the evidence of that? You won't be able to cite me an example. It's what you believe, I understand. No, you gave me two creatures that exist in the fossil record, both of which are extinct. That's all.
I cited it in the chat.
The last example, brassica and so on.
What, I'm sorry?
I'm talking about cabbages and kale and so on. I'm showing you how you can
cabbages and kale.
One.
Right, those are not anatomical difference, that's morphological difference. That's how you shape the same tissues that exist in the creature and shape it. But that's not a new feature with a new function.
Different things. That's it.
No, that's not a new feature with a new function.
What?
I'm sorry. It's not a new feature.
Different folds is new feature.
So now you need a new function. Listen, go ahead.
Changing the shape of the broccoli does not make it have new features. There's no example of a new structural design, a new anatomical feature with function arise in broccoli or cauliflower. Those are morphological changes. They can be more profound in plants than they can in animals, it seems.
That's already there.
But
every time.
That's your best example, is a morphological change in broccoli? What about animals? Can you cite me any example in animals? Plants seem to be more plastic than animals, you know.
Right, in some cases they're more plastic. A new morphological feature in animals. Oh my gosh, I'm so.
No, not morphological feature. Anatomic, not a new morphology, a new anatomy.
Okay, so, you aware of the same lizards you and Jackson Reed were talking about?
Yes, the cecal valve, which is an enlargement of an already existing muscle in its gut.
That's it. So every new structure is a modification of an existing one. That's what I'm trying to tell you.
Well, that's your philosophy.
No, that's just how.
But even in the lizards, that doesn't show a new anatomical feature. They already have the muscle. It just got larger and more pronounced.
That's how everything works.
Well, then you're saying that every existing anatomical feature is comprised of the same tissues?
No, you can change cell type. You can change.
Now you've got to have cell specialization go on in the genome during fetal development, and that needs genetic information.
Not really. You can have.
Yeah, really.
Well, epigenetics can cause change in cell type.
So you're going deeper and deeper into your philosophy, but you're not citing an example of an anatomical feature arising and in the lizard, it already has the muscle. A bigger muscle is not a new anatomical feature. Okay.
That's how it works.
I can work out with weights. Well, that's your philosophy. I can work out with weights and get bigger muscles. That doesn't mean I have anatomically changed. I've changed morphologically, not anatomically.
That's how the structures begin. They begin by modifying what's already there.
Well, that's your philosophy. But you can't explain a new structural design, a new anatomical feature arising by modifying the shape of an existing one.
So what about the cecal valve? That doesn't count.
The cecal valve is a muscle in the gut of the lizard. It already had, and it got more profound, more pronounced, larger, and their stomachs elongated. They became a bit longer, and their jaws became a little wider.
Okay.
Right. So those are morphological changes, not anatomical.
Therefore, to go from an ape to a human, you don't need any new morphological.
That's not true. Humans and apes don't have the same number of bones.
What? Maybe bones? But there are different.
There's a different number of bones in the skull of a chimpanzee than a human being. There's a different. Chimpanzees have a bacula. Human beings don't have a bacula.
Well, that's human being. That would be a loss. Then you already accept losses. But the number of bones, I find this strange because you guys accept like time.
None of this is observable or testable or repeatable. This happens in your imagination.
Are you saying that the number of bones in an animal is impossible to change?
No, I'm saying that's an anatomical difference.
But it would be the same bone, right? Just another bone, as you said with the muscle in the lizard. It's just a muscle, another muscle doing.
No, new bone would be new bone would be an anatomical feature, new anatomical feature. Enlarging an existing bone is a morphological change.
Oh, wonderful. So,
but humans can't be related to chimpanzees anyway because none of the recombination hotspot locations in their genomes align.
I love your example there because, for example, there are different breeds of pigs that have bred pigs to have more ribs to get more meat, and that's new bone, right? So that should satisfy the claim.
Okay, so so they've caused pigs to gain new bones, and so how is that going to help evolution theory that they got a new bone? And how did the features of a pig arise? How did we get a pig by anatomical change? Try explain.
That's a separate question. That's a separate question altogether. Remember,
what scientific evidence is there of that?
If I add a new bone, that would be a new morphological feature.
That's an anatomical feature.
We're able to breed pigs with more ribs.
Mm-hmm. Okay.
More number of ribs. Okay. So that's a new bone. That's more bone. Satisfied your claim.
Okay.
The same thing happens with snakes and so on too, but I'm just doing it with pigs.
Okay. So you're telling the creature express a new homeobox gene that puts a new rib on it. Okay. So is this verification that ribs that firstly, is this verification that pigs have never had that many ribs in the past? Secondly, how does this help you with the idea that the pig itself came into existence when there was no pig, the scientific verification of that? Because even your example, getting more... Hold on.
That's a separate question too.
Hold on. Getting more of the same feature on the animal's anatomy won't ever give you a different kind of creature either. For example, take a fish. Adding more fins to the anatomy of a fish is never going to give you something non-fish. You understand? You're still going to have a fish. You can have a fish with 3 fins. A fish with 30 fins is still a fish. You're trying to say fish become amphibian tetrapods with legs, digits, and lungs, and completely different designed eyes, and walked around on land breathing with lungs on legs. See? You can't get that by adding fins, nor can you get a pig by adding ribs. You need to build everything in the anatomy of the creature. We don't know of any mechanism for this, and you don't. And the new genes that created the extra rib in the pig, there's not going to be any scientific evidence that those ribs genes arose. That's a varying expression of existing genes that already make ribs. They've been it's a duplication of a homeobox gene is what that is. So build a rib here. The gene, you duplicate the homeobox gene, and now you've got an extra homeobox gene that says put another rib in there.
And that gene is very plastic.
So you can't get a fish like this to become a land-dwelling creature with lungs by changing how many fins it's got. Neither do you get a pig by adding ribs. You understand?
What about fish that have feet that crawl on the bottom of the ocean?
They're an interesting design. God made some pretty amazing
were they
creatures.
original designs, or were they adapted?
Probably.
Which one?
Probably original creation. God has
How can you tell?
a tremendous. Well, I can't. Neither can you. But God has a tremendous imagination. But one thing we know is
it just looks like a modified fin.
So you think it evolved? Good. Now, what's the mechanism for that? We don't know of one.
It just has muscles in different places.
It has different anatomy than a typical fish.
Yeah, slightly.
Well, I think it's more than slight.
But slight changes add up.
Well, so you're welcome to believe that slight changes to genetic information create new anatomical features. What's the evidence of that?
We just went over that with the pigs.
No, you're talking about reproducing an existing anatomical feature. Human beings can be born with 6 fingers too, and that's typically not a good thing because the 6th finger is usually inept and not properly controllable by the human being's hand. But human beings can be born with 6 fingers. That's not evidence that human beings came into existence or human fingers came into existence by new genetic information arising because of mutation. That's just a duplication of a homeobox gene that says put another finger on there. You can't get a human being by adding fingers.
So just as the gene can duplicate the number that it produces, it can change the orientation, the length, the thickness, other features of the bone.
Yeah, now we're moving into your philosophy. It all changed so much that you get the human being out of something else. That's what you believe. But you can't even tell me about the mechanism.
Are those genes off limit for change?
You can't even show me the mechanism of duplicate genes creating new information that codes for new anatomical features. We don't have that documentation.
Here's a good illustration. If I hand you 2 Bibles, and I take one and I cut out little snippets of it, and I create a sentence out of it, and I cut out the rest, you still have that original Bible, but I've created a new sentence out of the second one.
Taylor,
you now have more information than what you started with.
Taylor, your philosophy is
that's just a demonstration of the concept.
It's one thing. Yeah, I understand your concepts are not scientific evidence,
No,
Taylor. Do you understand that?
Just tell me how I'm wrong about that.
Taylor, my concept. Tell
me how I'm wrong.
Well, my concept is this.
How is that not new information?
Well, Taylor, here's my concept. See, my concept is that God did this, and there it was. See, hold on.
Yeah, magic.
But your concept is that for what you just described. But here's my concept. There it was. See. Now, I can't scientifically verify that for you by showing you the mechanism for that. But what I can do is show you the overwhelming evidence that that's what happened. In fact, I have some very, very powerful evidence from genetics that that's exactly what happened. But you're not able to show me a mechanism which creates the new genetic information that codes for a new anatomical feature. No scientist has documented that. It's never been documented.
So tell me how I'm wrong with my analogy. How has that new sentence not added information?
because firstly, this would be a scientific observation if it were true. We should scientifically observe new genetic information arising. Secondly, the fossil record. Hold on.
That's not what
Secondly,
I asked.
examining the history of living things on the Earth in the past shows us it didn't happen, because the fossil record shows that it is absent of the transitional forms that would have been there had evolution taken place. See, so we don't have a record of it.
No, that's wrong.
And we don't know of no scientist has documented this occurring whereby new genetic information is created that codes for anatomical features. It's never been documented scientifically either. So when we look at the historical evidence for living things on the planet, and it shows us it didn't happen, and then we look at the scientific literature, and we find no scientist has documented this either in a genetics way. We just don't. We end up empty-handed. We don't have evidence for it. You believe it, maybe, but we don't have evidence of it.
So that doesn't answer the question.
I just did answer your question.
No, just tell me how I'm wrong about. Tell me how I'm wrong. How it's not a new sentence.
How
is it not adding to information?
What?
If I give you a Bible, and I give you 2 Bibles, I take one of those Bibles and I cut it up, and I create a sentence that occurs nowhere else in the Bible just by repasting and scrambling up the words. Then I give you back that sentence. Now you have a Bible, the original Bible, plus that sentence. That's new information.
Well, that could be. But now show us the Bible made that way. You're positing that the organism is made that way.
That's how duplication mutations
work. I understand your philosophy about it. But what we need is the evidence that that's taken place by comparing genomes, for example.
We've observed this happening.
For example, the Italian wall lizard, they could have easily sequenced the genome of the 2 populations. And what they would find, and what they did do, is sequence the mitochondrial DNA of them, and they found no difference. But they didn't sequence the genomic DNA. Okay, and the reason they won't do that is because they know when they do, they won't find a significant difference that we could point to and say, look, the genes that code for the cecal valve arose. They don't exist in this one, because they'd find their genomes are identical. And there was no new genetic information. That's why they won't do that.
Oh, Neff, can you hear me?
Yeah.
Oh, wonderful. Okay, so I'm just asking you if you would what really constitutes a new anatomical feature. So is the transition from a lobe fin to a tetrapod limb, is that a new anatomical feature? Would that count?
That would be a change to anatomy. A limb instead of a lobe fin would be a change to anatomy.
That's it. Oh, that's it. That's what I'm trying to tell you, that every new structure is just a modification.
That's what you believe. I understand that. We've been over this 1,000 times already. Okay. It's getting boring.
Right.
The simple thing is you can't provide evidence that your philosophy is science.
What would count as an example of a new feature on an organism?
What would count as? What would you believe is? Instead of being able to give the evidence for your belief, evolutionists do this.
But I'm trying to tell you, what you're asking, it's a malformed question. If you're asking something that evolution doesn't do, then you're not asking for evidence of evolution.
And what you guys are actually doing is trying to say, can't you see it in your imagination? Look and
No, that's
see.
That's not what we're saying.
Just imagine. This is essentially what you're doing. You're swinging a pocket watch and expecting me to believe it. I am getting bored with this, really. I asked for the scientific evidence for your philosophy that random genetic mutation builds new genetic information that codes for new structural designs in the anatomy of creatures, and you guys can't put forth one. So you're trying to schmooze me with your philosophy. It's not going to work.
I would say what would make me happy is if you took a bunch of, let's say, cattle and you placed them in lakes and let them breed in water, and they could never leave the water, and they built these new anatomical features that would allow them to be much more sufficient for living in water. That would obviously make more sense since they're confined to that type of environment. I mean, take for example, we have cattle and we breed them specifically for weight. That's all we care about. How much they weigh and how good they taste. We would love if they could be like elephants. We don't really even care if they stay cows. It doesn't really matter. We'll eat anything. They could be gigantic and they should taste like bacon. But yet we can't get them to really much. I mean, they've got them as fat as they can.
Yeah.
And when they get them too muscular, then they don't taste good.
Right, and they're unhealthy. Those gigantic blue cows that they've been mutants they've been making, incredibly muscular things, just profound. They look bizarre. They're such a freak-looking cow, and they're terribly unhealthy too. The animal is defunct. Its health is shot. So, if evolution were, why is it that anybody should believe that evolution is true after the discovery of DNA? I mean, we've discovered that DNA is a package of information that operates algorithmically, it's organized linguistically. Why are we to believe that undirected chemical interactions can produce information, algorithms, and linguistics? You've got to be able to answer that question if you think evolution is true. It has to have happened.
No, because
how is that possible?
Created DNA doesn't rule out evolution. That just rules out abiogenesis.
No, not because of because of the fact, Taylor, that it's information operating algorithmically and organized linguistically. That's the proof of creation.
Creation doesn't rule out evolution, though.
Well, creation, Taylor, that's a nonsense statement. If creation is true, evolution cannot be true.
No, that doesn't follow.
There's old Earth creation, is what I mean.
Right, and they're just as wrong as evolutionists. Any form of evolution cannot be true if life is defined by information, algorithms, and linguistics.
But that information changes.
If theistic evolution were true, the evidence in the fossil record would show transitional forms.
And it does.
No, it doesn't. We've been over that. So how can evolution be true? By natural, undirected chemical processes, since what creates a feature in a living thing is information that's organized linguistically, operates algorithmically. And let me give you something let me give you something to really think about. Okay, really think about. The information in the DNA is overlapping and nested. Okay, that means let me explain this to you. I want you to have a jaw-dropping, oh my God, how do I answer for evolution moment in your mind. Okay, so I'm gonna share something with you that's going to give you one, whether you acknowledge it or not. Let me imagine that you have a sequence of information on the left. You got a strip of paper that's 5 inches long, and a strip of paper on the left that's right is 5 inches long. Now you overlap them. You lay one across the other so that there's digits on them. Okay, you align the digits, and what happens is now you take another strip of paper and it has digits on it, and you lay it across both of them where the joint is. So now you've got a third. Okay?
Gene overlap?
So here's what happens is the digits on the digits in all three pieces of paper is shared as sequences of information in those three pieces of paper. I can read from that just the letters that are on sequence paper 1. I can read only the sequence of information on paper 2, but I can also read 3. And now if I want to, I can read part of 1 and over onto part of 3 and create my own fourth piece of paper. So the base point I'm making is any given base pair, any given nucleotide along the DNA molecule is likely shared by more than one sequence of information at the same time. Now here's the interesting thing where it gets even worse. There are codes written upon codes in the DNA molecule that share base pairs going down the length of the DNA, and if those codes tell the machinery of the cell how to express the information under them, using some of the same base pairs as the sequences. An example of this might be, let's say I'll use the Bible as an example. The Bible says, in the beginning God created the heavens and the earth was void and without form, and darkness was upon the face of the deep. Okay, now that's a sequence of information that we have. That's actually 2. So we've got this sequence of information, but now I pick words out of that and I write another sentence that says darkness was upon, or I pick one that says, in the beginning, darkness was upon the deep. See what I mean? Just picking certain words, I can create a sentence out of those by using some of those same ones. And now what happens if that information that I'm just created by plucking out certain words in that tells the reader of the book how to read the rest of the book? This is what we have in DNA. DNA, there are codes written on DNA over sequences that share some of the base pairs. Maybe the base pair number, this one, skip 5, and it uses that one. It uses the next one, and the next one, and the next one, and the next one, but then it skips 3, and it uses that one and that one. And it skips 20, and it uses this one and this one. And it skips 100, and uses that one. That one skips 2, uses that one, that one, that one, that one, that one, and that one. Skips 10 and uses that one. Skips 20 and uses that one, and that one.
No,
You see what I
okay,
mean? All right, you understand what I'm saying? Now those base pairs, just those, all right, just those.
Listen for a while.
Create a code that say use the other bits that I'm also built of and do this with them. Okay. Now here's the problem for the idea of evolution building the genome by mutations and duplications. How in the world is it plausible to believe that you can get the regulatory information that shares the same base pairs with the DNA sequences that code for the proteins to be created at the same time? Because if you think that the genome was built by evolution first by just duplicating and adding raw information sequences, duplicating genes that are not yet part of anything, you're breaking the codes that tell the system how to use the codes below it. There is none. You've broken it. Or there is none there. Okay, so when evolution would have to be the magic of creation, in fact. If evolution were true, it would be creation, because it would mean that the building of the genome by mutation events would have had to have created the new sequences for proteins and whatnot in the genome, and at the same time built the codes upon them that share some of those same base pairs going down the length of it that tell the system how to use the codes for the proteins, and both are using the same bits of data. You can't imagine a process that can do this. Evolution is nonsense. It's impossible.
This sounds like irreducible complexity.
It is irreducible complexity.
Irreducible complexity. Okay.
So explain how that's logical. Can you give me a logical scenario whereby evolution could do this, continuously expand the genome with new codes for new proteins, and write the code that tells over the top of them, using some of the base pairs in those same codes for the proteins, how to utilize the protein codes underneath it? Can you explain how that's possible?
Okay, so off my head, I can't explain that for you now. I'm being honest, I can't explain that for you now. To refute those claims of irreducible complexity would take a lot of research, and most of the time they're fallacious, meaning that they look at the current state of a system.
Right, you just can't explain it. There's no way it can't be explained. Matt, were you about to say something? It's getting late, guys. I'm going to have to shut this show down. So,
right, what I was saying is that irreducible complexity is a description of the current state of a given biological system. Usually, what they do is to look at the system currently and try to remove a part and see if it will function. That's not how you test it.
Okay, so here comes your philosophy. It wasn't always irreducible in the past, right?
Right. Now you're getting
right, your philosophy.
Right, good. It's not philosophy.
Hey guys, it's Standing for Truth coming to you live tonight. I've got a guest with me. His YouTube channel name is Snake Was Right. We're gonna have just a little dialogue here on creation, evolution. Should be fun. I appreciate Taylor. I'm gonna refer to him as for joining me in this discussion tonight, as well as I've got Landon here, who's going to just moderate for us, make sure that the discussion is flowing nicely, and just making sure that it's going well. So I want to introduce Taylor. If you wanted to say a few things, brother, go ahead. Floor is yours.
Howdy. My channel name is Snake Was Right. and I'm an atheist. I'll mention that I went to a Catholic school for 4 years, just for that background. That's pretty much where I became an atheist. And the channel name, just to explain that, Snake was right, just means I think that the knowledge of good and evil is a good thing to have. So that's my intro.
Awesome. Well, I appreciate that. Landon, you'll be moderating for us. We're going to go, so structure-wise, it's just going to be free-flowing dialogue, little interruptions. Landon's going to be moderating, making sure everything goes well. After the hour mark, it can become a free-for-all at that point if people just want to talk about random things or whatever for another 45 minutes or an hour. Should be cool with me. Thanks for moderating for us, Landon.
Oh yeah, no problem. It's good to be here. So
awesome. Thanks, brother. I appreciate it. So I guess my question would be just to kind of start it off, Taylor, because I'm sure we'll be throwing around the word evolution quite a bit during this discussion. But at the end of the day, I think the term evolution can mean many different things, right? In general, it just means change over time. I would say, biological evolution would mean a change in, say, allele frequency or gene pool in populations over time or generations. Like, what would your definition be?
I think that's a fair definition for biological evolution.
Okay. I think for me, in regards to, say, the model that I hold to, like a biblical based model, I'm not opposed to change. I'm not opposed to, a change in allele frequency or gene pool, of course, but that type of, micro evolutionary variation, Taylor, where you get a change in, say, the frequency of the expression of different traits. I don't think that's really disputed among, creationists or evolutionists.
Mm-hmm.
I just think that explains, the small scale variation, but not the major innovations, the major origin events of, say, major new forms of life. Do you think that allele frequency, change in allele frequency, would explain, say, quote unquote, the arrival of the fittest? Go ahead.
Yeah, that's what the point of the theory of evolution is to explain the biodiversity. And that's what Charles Darwin's book was, The Origin of Species, speciation. And so, yes, I do think that there is sufficient evidence to say that the mechanisms of evolution, whether they be just mutation or just changing allele frequencies that already exist, or dialing up the tallness or shortness, of whatever trait, we do have evidence of that creating speciation.
So would you say, just based on what you said on the mechanisms, would you say that, you believe in an unguided process of natural selection acting upon random variation or random mutation? Like, would that be the basic mechanism behind that type of large scale evolution?
Well, it's not unguided. There are evolutionary forces acting on the populations at all times and maintaining the allele frequencies or getting rid of unbeneficial ones and conserving the beneficial ones.
Right. So I guess it would be my position that, say, if we're looking at mutations, I would say that mutations are more so destruction and not construction. I would say that these mechanisms, neo-Darwinism, I would say that it lacks, incredible explanatory power in regards to, the origin of phenotypic complexity, anatomical novelty, and also, the origin of non-gradual modes of transition, say abrupt fossil appearance. I think, in short, I just think mutations and natural selection, natural selection is a fine-tuning process, and you can certainly get variation based on mutations, but I would say that they lack significant creative powers. Like, what would you say to my point on that?
So I had to listen back on my debate with Nephilim a couple of times to understand really what he was trying to say. he kept asking me, how does the beneficial effects of a mutation outweigh or overcome, is what he said, overcome the deleterious effects. And so it took me a couple of times to actually understand, because I think what he was actually trying to ask is that, well, I think what he was assuming in his question is that each mutation, no matter if it's beneficial or not, that it necessarily comes with deleterious effects. And I was just trying to explain that while most mutations are harmful, there are some individual ones that are not. And those individual ones they could carry a trade-off, but sometimes they don't. And it's not like you carry with you all of the bad mutations. And I think that's what Nephilim was assuming there, because you can just get a single beneficial protein out of a mutation, and you don't necessarily have to get bad mutations. It's just a numbers game.
No, I appreciate the response. I think, just to clarify, would you say that mutations that are beneficial and have no ultimate deleterious effect, would you say that they would be rare, more so?
Very rare.
Okay, so I think, so if I look at my starting point, say in regards to a biblical model and the fall, so we would predict death, decay. So obviously, judging by the first few minutes of our back and forth here, we don't disagree on mutations. We don't disagree on natural selection. But I think the bottom line would be that, selection would remove, and I think you agree with this based on listening to what you said, selection is going to remove the worst deleterious mutations, the worst detrimental mutations, and it's going to amplify only the best rare beneficial mutations. But my point would be that this means that, say, the accumulating damage based on these, low impact deleterious mutations, the one that natural selection, the low impact mutations that are invisible to natural selection, that accumulating damage is largely invisible. And to make it simple for the audience, kind of like rust on a car. Because if you think of it this way, Taylor, we've got a genome of, say, 3 billion letters. And if you take out one letter, change one letter randomly, say, are you going to have a huge fitness effect? Obviously not. It's going to have a tiny fitness effect. And
it could knock out the whole system.
Well, I would say it's kind of like rust on a car. You can't see each rust event, but it's continuous and destructive. So
if you get rust on the right part, the whole thing won't work anymore. But you could get rust on somewhere where it doesn't even matter.
Well, the thing is, if you're looking at the genome as a whole, and just say, one random mutation that might not have an obvious, phenotypic effect on the actual genetic content, it's going to be slightly deleterious, kind of like that individual rust spot on the car. So let's say the accumulating damage is largely invisible, like I was saying, but these adaptations, obviously I'm not disagreeing with those, say antibiotic resistance, melanoprotein example, like whatever it is, those will be highly visible. But that means you're going to have to present me with, say 1,000 examples of adaptation through whether it's beneficial duplication, point mutation, just to address the key. I think the key issue, Taylor, is net gain versus net loss.
Yeah, I think I see the problem here. And I do want to point out that mutations are very frequent in bacteria and viruses.
Right.
And we can see them evolving rather quickly. But your point about the genome accumulating damage is it actually is able to edit out things that it doesn't need or that are damaging it. And there's just like as many ways as it can acquire beneficial traits, there are like at least dozens of different ways that the organism can get rid of bad traits. So for example,
yep, go ahead.
Well, the genome is constantly degrading in a way.
I agree.
It's always victim of entropy, but the good genes will spread. So the genes that don't get used, that have no pressures on them, will pretty much just get forgotten in the shamble. and the good genes will keep getting used and keep getting spread. And the deleterious mutations or genes will just, sometimes they'll just disappear due to mutations. There are gene deletions that are mutations as well. And so there's just so many ways that the genome naturally loses this information, but it will conserve the good information. And one of those ways is breeding. That's one of the great things about sexual reproduction, is because you might have a good gene and a bad gene, and your partner might not have either one, and you have 50-50 chance of passing on. You might only pass on your good gene, or you might only pass on your bad gene. But if you pass on the good gene, then that's going to conserve in your child, and your child might not inherit your bad gene. And that's how, through breeding, through that is a selection process of just reproducing.
Well, okay,
The bad stuff just naturally falls away.
Yeah, I would say natural selection is just a fancy way of saying, differential reproduction, who's reproducing the most. But I wrote a few things down on what you said here. So, for example, you said beneficial mutations are rare and the genome is constantly degrading. And we see entropic degeneration on a genomic level. So I agree with that. But I think it's pretty obvious that due to the rarity of say beneficial mutations that are actually not deleterious to some extent, those beneficial mutations are not going to compensate for, as I said, I think it comes down to a net loss versus a net gain of information. Those beneficial mutations are not going to compensate for the net loss of information. And natural selection, because you talked about traits, natural selection, it's a fine-tuning mechanism. It keeps the species as good as it can be. So I don't disagree with that. So we do agree on that, but it's severely constrained by, what's called selection interference. And just to go into that a little bit, selecting for one trait, as you talked about, interferes with selecting for another trait. And when you have billions of traits, Taylor, segregating in the population, then the selection process actually starts to work against itself. And what that means at the end of the day is that you end up with being able to only select the best and worst mutations, as I talked about earlier. So I think evolutionists, I think you're guilty of this right now, they assume, based on universal common ancestry, right, it's got to be true type thing. So they're assuming that these mutations, they're going to have a net neutral effect. But we know that's wrong based on this selection interference. And I think, just to put it in a nutshell on everything that you said there, and I wrote down, the problem is that, we know that beneficial mutations are very rare, which we agree on, so that's good. And non-neutral mutations are consistently deleterious, as we should actually expect, with typographical errors in a text. So I think increasing fitness is going to be very difficult. And very much problematic. And you talked about, beneficial mutations, say in bacteria, or just beneficial mutations in general. The problem that I have, though, Taylor, is that, the best beneficial mutations, they're mostly reductive. Let's take sickle cell anemia, for example. It's got a significant impact. I heard you talk about, that with Neph, and I agree with everything you said. But what it is, it's a broken gene, broken protein, and in the long run, it's not really taking things forward, and it's not really addressing the issue of net gain.
Broken is relative. There's nothing broken in the chemistry. You can't break chemistry. It doesn't understand what it's doing. It just does what it does. So when you have sickle cell, it just doesn't work the way that you want it to, maybe for oxygenation, but it has other beneficial effects.
So you're saying it's deleterious in one way, but it's beneficial in another. But I'm saying it comes down to a net loss versus a net gain. So how are you going to compensate for the information loss based on, a couple rare beneficial mutations? Because if you're throwing out a ton of information, and you're seeing erosion of information, erosion of nucleotides, how are you going to compensate for the actual information loss? Because a couple of rare beneficials are not going to counterbalance the accumulating damage. So with me, under the assumption of death, decay, degeneration, that's consistent. But to take your fish to fisherman or your bacteria to biologist and build a genome, I don't know how you're going to build a genome by degrading information. And you talked about bacteria. I just want to touch on that real quick, see what you think. You can just Google, bacteria, reductive evolution, and it's a commonplace. There's tons of papers that, they're subject to reductive evolution. Like if you take Lenski, for example, there were beneficial mutations, there were adaptations seen. So I'm going to agree with you on that, but they were largely accomplished, Taylor, through loss of function, loss of promoter, loss of genes. So his own data, Lenski, for example, he's revealing clear evidence of genetic degeneration. And that's not really going to take your fish to fisherman. It's not going to take your evolution forward because it's a net loss of information. So my question would be for you, I'm really curious, how do you deal with the net loss versus the net gain? Go ahead.
All right, well, I'm going to need a couple minutes. Well, you mentioned loss of information being a mutation, and that loss can actually be a gain. So we have a bunch of DNA, or we don't have a bunch of DNA that the apes do, and that's one of the major differences between us and them, is we deleted some regulatory mechanisms. So you can actually have, that is a way of getting rid of bad DNA. We deleted a bunch of DNA and got smarter as the result. So,
like, I just want to clarify, just to follow what you're saying. Is this more hypothesizing, theoretical, because you're looking at both the genomes and comparing, or is this something that you've actually observed and seen, say, in the lab?
Well, when you compare the two genomes, there are similarities, and there are genes that we don't have that apes do have. So if you put that DNA, if you took the ape DNA and you took out those sections of DNA, I could send you the paper if you need that, but you would probably get a human. We can't clone humans, so we can't actually test that. But we would have
kind of theoretical
the same genome. Well, you would be working with a human genome at that point. You could just insert that.
I get what you're saying, but you're not really, like, let's say observable right now, based on what we know about natural selection, based on what we know about, mutation accumulation, because it's largely recognized that we accumulate, 100 to 300 new mutations per person per generation. That's 7 billion mutations this generation, given the population that we know.
All those fine-tuning processes and mechanisms, they work against deleterious mutations and even useless DNA. We lose stuff naturally. If you don't use it, you lose it, basically, is what's going on here.
No, it's because these low impact mutations, as I was talking about earlier, they build up relentlessly because these deleterious mutations, Taylor, they're pouring in
and they delete themselves.
They can be removed,
right? And they don't get passed on as frequently as beneficial mutations.
No, they do, because most mutations are not neutral. They're nearly neutral. They build up in the population in the genome. They're invisible to natural selection. So how are you going if we inherit a multitude of old mutations?
Because if you don't use it, you lose it. If there's no positive selection pressure, then it's likely it's just randomly going to go away one day, and that happens.
Well,
those are gene deletion mutations.
Here's the problem, because, and I think you agree with this, all the papers say it, human mutation rate is 100 mutations per person per generation. That means, Taylor, that our children have about 100 mutations more than we have, and our grandchildren are going to have 100 more mutations than they have. So on a population level, like I said, that's 700 billion new mutations entering the human population this generation. So the question I'm asking you is, what type of selection could eliminate so many mutations that are pouring into the human population? Because they are passed on, and if they're at neutral, near neutral nucleotide sites, they erode information, but selection does nothing about it, because selection acts on the phenotype, not the genotype. It's either, the organism as a whole or not at all, correct?
No, because we actually split our genotypes when we reproduce.
Correct. But those mutations, the ones that we're inheriting from our parents, 100 to 300, those are coming in to the next generation. And if they're near neutral, selection can't do anything about them, because I said at the beginning that selection will amplify the best beneficial mutations and get rid of the worst deleterious mutations. But it's those low impact neutral mutations, like rust on a car or a typographical error in, say, a book the size of an encyclopedia. Those are the ones that build up and erode our information systems over time. And there's no way natural selection can get rid of those. I mean, how do you explain that?
Natural selection can get rid of any gene. If they're near neutral, that it's not really doing anything. There's no selection pressure toward it other than just pure entropy. So
Exactly.
if it becomes a problem for the organism, then there is a major selection pressure and it will get selected out.
No, because
or the organism will go extinct, which most organisms have. It's that rare one that ekes by that will continue on the line.
See,
and
it's because
populations have bloomed and shrunk, and almost all of humanity was almost dead at a certain point, and we're back.
See, it's because these mutations at these near neutral nucleotide positions, Taylor, they're deleterious and subject to random drift, which makes them immune to selection. But these nucleotides, because you're saying it's not going to do anything because it's near neutral, but those nucleotide sites, they contain meaningful information, and the mutation contributes to the erosion of that information. So collectively, since we agreed that beneficial mutations are rare, obviously the near neutral nucleotides are going to account for most of the information in the genome. So just to make the audience understand it more, to put it into an analogy, this would be as true as the fact that all the seemingly insignificant letters in a book collectively add up to a clear message, Taylor. If we start to introduce typographical errors, say, most of the individual errors, they're only going to have an extremely trivial effect on the total message. So individually, I agree, they're truly insignificant, as you said. But here's the issue. It's that if this process is not halted, the message will eventually become corrupted and will eventually be completely lost. And that's what we're seeing in our genomes, because natural selection can't do anything about these near neutral, low impact deleterious mutations. So once again, how do you explain the net loss versus the net gain if you're going to take your fish to fishermen? Go ahead.
Well, since you haven't observed any of that, that's a much bigger leap than any amount of changes proposed by macroevolution.
Well,
we
are you saying
we haven't seen anything die just because it's so genetically corrupt? We can do that by breeding like dogs, but they're still pretty much healthy. And if you start breeding them with other dogs, those deleterious effects will start going away because they're mixing with healthy dogs.
The thing is, if you look at a lot of this dog breeding, let's just say, like a wolf to a Chihuahua, for example, for the most part, that's the degradation of information. If you take all the Chihuahuas and throw them back into the wild, obviously they're going to last one day.
It's not degradation.
Well, you're losing a lot of information, like the allelic variety and the allelic potential in, say, something like a Chihuahua versus something like a wolf. Obviously, we can agree that
they're essentially the same species. They can interbreed.
Oh, I agree that they can interbreed, but the allelic potential and the allelic variability in something like a wolf. I mean, I've talked to dog breeders, and they can take two generic dogs and probably breed almost anything. But if you take two Chihuahuas, well, there's not enough allelic potential there to breed really anything else, because the information has been lost. But I want to back up what I said about
Chihuahua out of Chihuahuas. You could just, because there are Chihuahua mixes, you could just keep mixing that whatever has Chihuahua in it with a dog of another breed, and generations down the line you will have no Chihuahua.
Well, obviously we're talking about artificial selection here, but it's still not explaining, you know, the net
Same
loss
mechanisms.
the net gain, because there's two models here, right? Like I'm predicting death, decay, degeneration, and that's exactly what we see. But you know, to go from your single cell, multi-cell fish, amphibian, reptile, mammal, you know, monkey, bird, whatever it is, you know, that's going to take massive increases in genetic information. And if there's a net loss versus a net gain, and what we see is a net loss of genetic information based on the accumulating damage of near neutral low impact mutations, that pretty much falsifies that type of, you know, pawns come to people evolution. And I got a ton of examples because you talked about extinction, Taylor, and I've got a paper right here that talks about mammoth populations. So paleogenomic evidence, it's in the journals, but I can send it to you. But these mammoth populations were highly had a high genetic load, elevated genetic load, which led to their extinction. And even Neanderthals, they were highly inbred and had a very high genetic load. And the paper talks about how they were 40% less fit than modern humans based on accumulating genetic damage. And exactly. So all we see is death,
So they don't
degeneration,
get to pass on their
extinction.
shitty genes.
But the thing is, they're dying due to accumulation of near neutral mutations. And a lot of them are dying because they're highly inbred. So they're subgroups, they're mini populations. So what that is, it's a sneak preview as to where we're going based on this accumulating genetic mistakes. Because we know if it's a small, you know, inbreeding population, well, they're going to decay and go extinct sooner. But the point is, it's inevitable. And natural selection can't act on these low impact deleterious mutations. So, you know, once again, how do you explain this net loss versus net? Are you familiar with the hobbits, Homo floresiensis?
Yeah, so you've already demonstrated how natural selection takes care of this, because there are other animal lineages living on past all these other animals that have supposedly died of genetic corruption. But this is how natural selection works. 99% die and the 1% goes on.
But the thing is, the 1%, as I said, if we're accumulating 100 to 300 new mutations per person per individual, we're carrying along those near neutral mutations. So we're only declining with age, because there's a pandemic. You could probably even agree with me, Taylor. There's entropic degeneration on all levels. Just look around. You know, Alzheimer's, dementia, autism, asthma, autoimmune disease, cancers, immunological related disease. I mean, it's just mutation related negative effects everywhere. So I don't see how that's consistent with your
well, how are these diseases mutating to be more fit?
Well, they're not mutating to be more fit, because the accumulating genetic load is what's revealing these. Because, for example, let's take
better at killing us. Some of them.
I'm sorry, go ahead, repeat that.
Some of these diseases are getting better at killing us. We can barely keep up with the flu. We now have MRSA, which is it's resistant.
Yeah, I agree. But the thing is, disease, death, decay, degeneration, mutation, that's not going to that's more consistent with the biblical based model. And even Lenski's own experiment with the so we see it in, you know, in bacterial populations. And you mentioned
a problem for genetics anyway. That may be the case, but there's no papers that I'm aware of that puts any kind of clock on it, because these 100 mutations per generation is, if they're near neutral, they're just not going to impact the organism at all. So it doesn't matter. As soon as they become subject to selection pressures by being beneficial or not.
Well, I've got a paper as well, and what it shows is that even if a substantial fraction, Taylor, of the human genome is functional, say is not junk DNA, then the evolution of man would not even be possible based on this genetic degeneration. And because I know a lot of the evolutionists like to fight, and I'm sure we can get into that. They like to fight, you know, ENCODE and how they revealed that, you know, over 80% of our genome is transcribed into RNA. But even if I just gave you, if I just said, hey, listen, for sake of argument, most of the genome is junk. In this paper, it demonstrates that human evolution would still be impossible even if the genome was only 10% functional. And like I said, ENCODE has obviously revealed that over 80% of it is transcribed into RNA suggesting function. So once again, I mean, this type of evolution seems to be falsified, impossible, and more consistent with like a biblical-based model of death, decay, degeneration. Go ahead.
Could you cite that paper?
Yeah, it's here. I'll put it to the side. So, are you? Are you familiar with? I'm gonna put in the side chat, brother. But I wanted to ask you. Are you as you look at that? Are you familiar with? Well, you mentioned viruses. Okay, so I've shown you, bacterial examples. I've shown you mammoth populations, Homo, Neanderthal, hobbits, for example. Entropic degeneration on all levels in regards to diseases. But even the H1N1 human version, and I got a paper here too if you wanted to see it. It's widely accepted, and you might be aware of this, that it went from, and this is the human version. They've got swine flu versions circulating right now, but that's besides the point. The human version, Taylor, it went from a red hot pandemic to a whimper to an extinction event in 90 years. And what was observed here in the study was a linear accumulation of mutations. So this is also genetic entropy at work. And evolutionists themselves, they cannot explain why the H1N1 virus is extinct in humans, because adaptive evolution obviously doesn't cause extinction. And the paper goes on.
It's not even a human virus. So you witnessed an attempt to evolve and it failed.
Yeah, this virus went from...
It attempted to adapt to a new environment, sort of, it worked, and then it couldn't go any further.
Well, what was seen was that there was a decline in functionality, which was predicted based on what we know about low impact.
It's not fit for human infection.
So are you familiar with what I'm talking about in regards to the H1N1 human version that went, because it was a red hot pandemic at first. Would you agree with that?
Mm-hmm.
I mean, it killed how many millions of people? But in 90 years.
All of this is consistent with evolutionary theory.
I don't understand how, death, degeneration, mutation accumulation, say bacteria getting rid of genes, short-term but long-term degeneration. I don't understand how that's gonna, because for example, you talked about gene duplications a lot, and we can get into that, but even gene duplications, Taylor, those are almost, and tell me if you disagree with that, but gene duplications are almost universally deleterious. So even if I gave you, for sake of argument, just to not go on about it for an hour or 2 hours, if I just gave you a few rare beneficial duplications, they're still not going to be able to offset the many accumulating deleterious mutations, let alone all the other accumulating mutations that are not duplicated related. So, I mean, how do you explain the net loss versus the net gain? Because I'm not being convinced of that type of large scale evolution based on degeneration and decay of a genome. I mean, that doesn't make any sense to me. Can you make me understand it?
It's just a numbers game. You ask enough girls out, one of them will say yes.
My wife said yes.
Same thing. You said gene duplication is almost universally deleterious, and that's just not true, because you can have extras of one gene. That's just basically a backup. Sometimes it's a problem, especially when the gene relies on a mount, and you can get too much of a thing being transcribed, but that's not always the case, especially with sex chromosomes.
Do you have, because I noticed you said numbers games, if you were to put some numbers on it, are you familiar with, well, you're probably familiar with Haldane's dilemma, but are you familiar with the waiting time problem?
I think so. I'm going to need a refresher.
Well, Haldane's dilemma is a little bit more old school, but it talks about, these mutations we're talking about, because we can talk about all day, duplications are going to build the genome, but it's all about these beneficial mutations if they exist. It's all about them going to fixation in a population. So there's a paper here called the waiting time problem, and I'd like to hear your thoughts about it, because you seem to know your stuff here. And what it does is it takes into consideration, Taylor, a pre-human population with a population size of approximately 10,000. And what we're looking at is how long is it going to take 2 nucleotides in that genome and then switch them to 2 other nucleotides at the same location. But if you're waiting for just one single mutation, Taylor, a single nucleotide, say, a single nucleotide change, and you're waiting for the change, because this is what it all comes down to, that change needs to take hold in the population and become selected to fixation. And it's very long to take that change and make a single specific nucleotide change, about 1.5 million years. So there's
I think I'm aware of this. If I'm not mistaken, it was a program written for a creationist that was commissioned for him, and he basically does not understand the model.
Well, the thing is, if you look at the,
I would have to look it up again, but that's what I remember of it.
Well, from if you look it up in the critiques, it's pretty rock solid. It's been read by over 10,000 scientists with no serious critique or rebuttal. But if you're looking at genetic words, say to get a string of 8 nucleotides, the time that it would take would be more time than the universe has actually had. So
what was it called again?
What's that?
What was it called again?
The waiting time problem. I think my whole point,
I remember researching it. I don't remember the exact details, but
it's a newer.
I was not convinced.
The thing is, what I'm trying to say is there's a multitude of problems. So I mean, you can go to the waiting time problem. You can go to Haldane's dilemma. Mutation, fixation. But the thing is, it all comes down to net gain versus net loss, and I haven't really seen any solid examples of, well, for one, how natural selection is going to compensate and how beneficial mutations are going to compensate for the influx of deleterious mutations. I mean, we're clearly going down and not up. And are you familiar with linkage groups?
Yeah.
So, for example, linkage groups, if those blocks of DNA are immune to recombination, for example, it's the best way I can say the good mutations and the bad mutations in these linkage groups then cannot be separated because they're immune to recombination. And as we agreed earlier, the bad mutations are overwhelmingly deleterious because the beneficials are rare. They outnumber the good mutations. So these linkage groups, aren't they only going to get worse and not better? I mean, they're only going to degenerate if these bad mutations are linked. You can throw in 1 or 2, say, beneficial mutations, but at the end of the day, they're just going to be overcome with bad mutations. It just seems like on all levels of our genome, it's just nothing but decay and degeneration, and that's what I personally would expect, given a biblical-based model. And it seems like the opposite of what you'd expect given a model of universal comprehension.
But everything shuffles around. Even the linkage sites are shuffled around. Every single thing inside the cellular mechanism shuffles around, and so you get a diversity of these combinations, and it's very, very, very slow. That's why evolution takes place in the millions and billions of years. So,
well, I mean, that's why.
So to go back to what I was starting off with, you shuffle things up and you get this diversity, and only the best ones get to go on. So it's actually a self-improving mechanism.
Right. So as I talked about at the beginning, I don't disagree with the fact that natural selection is going to amplify the best beneficial mutations, say antibiotic resistance, for example, just to name one, and get rid of the worst detrimental mutations. But it's the low impact, near neutral deleterious mutations that natural selection can't do anything about. But I want to say something about the recombination real quick, and then you can address that. In those genomic regions that I'm talking about that are linked, the whole point is that there is no sexual recombination. So the good and the bad mutations are inseparably linked, and they can't be teased apart. So those
linkage sites change, though.
They're still linked, though. I mean, they can't be.
But the sites of those linkages can change.
They can. Oh, I agree that they can change, but they're going to change for the worst. Those linkage groups are only going to degenerate. It's a downward spiral, and it almost guarantees net genetic degeneration because they are linked, they're not teased apart, and those regions are going to experience predominantly bad mutations, and they can't be separated from the rare good mutations. So you throw one or two, just for sake of argument, in there, they're going to be neutralized by the large numbers of bad mutations. So that just speeds up the degeneration process. So there's too much degeneration going on to actually build a genome. How do you build a genome based on a net loss of information?
As soon as you start affecting the organism's ability to survive and breed, that gene line dies, so that it edits itself out. So, for example, it minimizes the impact every time. Like for turkeys are a kind of cool example. So only there will be like a bunch of male turkeys. Only the biggest, best, most healthiest of the turkeys all the other males will fight him, and whoever's the best, they'll all be his posse, and they'll protect his territory for him. The weaker male turkeys don't get to breed. Only the strongest one does. So
right,
they're maximizing. So if they have a little bit of deleterious effects, they're still maximizing the one that has the least, even if it's just these little rust spots.
I would say
it's constantly selecting out the bad and selecting for the good.
I would say, just so we don't go in circles, I think based on, just to sum up everything here, I would say, and then I'll see what you think about that. I think it seems like you think as long as beneficials happen, no matter how rare they are, Taylor, this type of bacteria to biologist evolution must be true, because it seems like, as long as you can show me selective adaptations happen, which I agree. But the thing is, it's hard to grasp how difficult it is to build a genome apart from design, because a population, Taylor, it can be undergoing genetic, kind of like these turkeys, okay? Like natural selection, fine-tuning process. These populations can be undergoing genetic decline due to the vast numbers of slightly deleterious mutations that I'm talking about, even while selection may be amplifying a handful of these beneficial mutations. And to make it understandable to the audience, to put it in layman's terms, just picture a 10-year-old car, Taylor. It's degenerating in all possible ways. You can install new windshield wipers, change the brakes, switch out the tires, but it's still degenerating in all possible ways. There's been an improvement, I agree with that, but not the type of improvement that's going to reverse the systematic degeneration. So I think it just comes down to net gain versus net loss, and I think the evidence is more so on the side of someone who holds to a biblical-based model. I don't understand how you're going to build a genome based on everything we've been talking about. Go ahead, brother.
So the mutations don't know they're beneficial or deleterious. Like, let's go back to H1N1. You could describe their leap to humans as an environment as a beneficial mutation, because it allowed them to leap from one habitat to another. But apparently that wasn't a good. Well, it is what it is. But once they were in the human systems, they found that they couldn't adapt much. So they did change, and they couldn't adapt, and they died. And what you're describing happens a lot of the time. But then there's some of the cars come out without rust spots, and they continue on.
Well, I mean, you can't see each rust event, but it's still continuous and destructive. And in the H1N1 virus that you're talking about, from what I'm saying, that would explain why the mortality rates went down, because we agreed that it did start off as a red hot pandemic, went to a whimper, went to an extinction event. But what they seen was that the virus started off more human in its codon usage, and then it ended up randomized. So what it did is it actually became worse at interacting with human DNA over time. And if it's using the wrong codons, Taylor, that means that it's going to be less efficient at replicating in human hosts. So that's exactly what's expected under a model of genetic entropy, but that model is predicted under an assumption of a biblical-based model. So I mean, isn't that consistent with the model that I hold to, Taylor?
It might be, but to go back to your like rusty car analogy.
Yeah.
So if this car just accumulates rust over time,
right?
Basically, to keep it analogous to biological evolution, you would have to have a car that was able to adapt and incorporate rust into its system. That's all it does. It just, all right, I'm rusty now, and it works with it. Right. Because rust to the cell, or damage, you would say, to the cell, it doesn't see as damage. Those are just the new conditions. So it can adapt to that. Rust on a car, you might think is bad, but if the car understands how to use rust, then it's a better car now in every way.
Well, I mean, I've seen 20-year-old cars that are still driving. You can get them safety, take them to the mechanics. They'll do what they can do. They're all rusted out, but that car is probably not going to last another 3 to 5 years. So sure, it's driving under the capabilities that it has, but like I said, just like each rust event is not seen.
The cars don't replicate, so. The analogy only goes so far, which is why I just described a magical car that could incorporate rust. But what cells do is incorporate what they have, and they just make random combinations and random changes, and it's slow, and it's it's 99% unsuccessful. But the 1% that does succeed ends up spreading all over the place.
But the thing is, these, like, even with genetic drift, these low impact deleterious mutations, they're immune to selection, and that's the problem. I think my position is just simply, these rare beneficial mutations that we're talking about, they cannot and will not compensate for the relentless influx of deleterious mutations. You're saying that they will, but you said earlier that this process of evolution, natural selection acting upon random variation, random mutation, it happens with lots of time, billions and billions of years. So that's why I say the evolutionists, they don't actually have a science. They hope, they dream, they imagine, but that type of evolution, Taylor, it really only happens in the imagination. It's kind of a faith, in my opinion, it's a faith-based science fiction religion, because it goes against everything that we know about empirical science. Because we just spent almost an hour talking about the degeneration of our genome, and you're saying that a few rare beneficial mutations, which
you can't tell me how much time we have left, though. So that does not go against the theory of evolution, because we can have both. We can have both evolving and devolving at the same time. We can have this long history of change in biodiversity, but they're still accruing these miniature mutations over time. We all might have it. That's unconfirmed. As far as explaining, I'm sorry, I lost my train of thought on that, but.
Well, are you familiar with the research? I mean, but we might just have to agree to disagree. I think I would like to just point out, but before we might switch topics slightly, I've pointed out extinction events in, obviously the H1N1 human virus, reductive evolution in all types of bacterial populations, extinction events based on increased genetic load in mammoths, populations in Homo Neanderthals, the hobbits. There's evidence that Homo erectus, Homo naledi experienced reductive evolution. We see it in cheetah populations, butterfly populations, and we see it in tropic degeneration, a pandemic in regards to cancers, autoimmune, immunological, asthma. I mean, we see it everywhere. And I think it's obvious that degeneration, the loss of information, is not going to take your fish to fisherman. And a few rare beneficial mutations is not going to counterbalance the influx of deleterious mutations. So that's my position. If you want to say a couple last things, because I think at that point we're just going to agree to disagree. I think the evidence is on my side, and I think the evolutionists have to do a lot of imagining to ignore all that evidence. But what do you have to say to that, brother?
Well, it's interesting then that most of the scientists would agree with my conclusions, or rather, I would agree with theirs. But so the science is on the side of evolution, because your main criticism is that we weren't actually there to observe every specific little change, and we're just kind of assuming based on what our model would tell us, and that's just it. The model is based on mechanisms that we've proven in the laboratory. All these cellular mechanisms for selection, or for how the genes work and how the genome works, and all the selection pressures in natural selection. We know all these things happen, and we know how they happen exactly down to the molecule. And so when we have this theory, and it can explain things that we don't know. And while we didn't observe it, this is precisely how we solve crimes. If we go over, we find a dead body, there's a gun next to it, and there's gunpowder. We never saw anyone get shot, but we see a dead guy with a bullet in him. There's gunpowder. We know how guns work. It ejects the gunpowder and the bullet when it's fired. So it's the same thing with all the science.
Well, and that's why I say, the evolutionists have a nice science fiction, fairy tale on their hands, because I would say time is the god of the evolutionists, because you have to dream, you have to hope, you have to imagine that the degeneration and decay of prior information is actually going to take your fish to fishermen. So to look at this crime scene you're talking about, if what we know on an observable level is that these low impact, near neutral, deleterious mutations are actually destroying our information systems, but then you want to look back and conclude that your amoeba-like ancestor turned into a whale over billions of years. I mean, that's going to be incorrect.
I don't want to conclude it. But we look at the biodiversity, and what we know of cellular mechanisms explains all of the biodiversity, unlike creation, because there is no explanation for God having created wasps that sometimes eat fruit, and some wasps that can only replicate by laying eggs in the brains of beetles. That is fully explainable by random mutations and natural selection.
Well, if we look to
why whales have hind legs inside of them.
Well, those
why would God give them that?
Well, those hind legs, those apparent vestigial organs, are now known to be highly functional and important in sexual reproduction. But I think that type of evidence is just based on an ignorance of. So I'm not saying you're ignorant of science. I'm saying, like, in the past, what they assumed was useless, functionless type organs, and now through further scientific investigation. So I think in the past, for example, they had over 140 vestigial organs. And now, I mean, we can sit here and argue over 2 or 3 possibly, but due to scientific advancement, and even ENCODE, I'm curious your thoughts on that. They found that at least 80% of the non-coding DNA was transcribed into RNA, suggesting function. So the trajectory of discovery that we see now, based on the accumulating papers that discuss new functions in these regions, favors genome-wide functionality. And one other thing I want to say is, we're just beginning, Taylor, to understand the developmental process. How coding and non-coding DNA coordinate when, where, and how, say, structures are sequentially put in place to produce a fully functional organism. I mean, in 9 months, a single-celled embryo into a fully human being. And comprehensive knockout experiments for a mammal, I'm sure you're aware of this, have not yet been performed. So that chances of genome-wide functionality are pretty strong. So where's the evolutionary leftovers from this process you're talking about? Because I think that goes right into the vestigial organ argument. I mean, do you agree that over 80% of our genome is functional?
Yeah, for most of it is non-coding, but a lot of the non-coding stuff has a regulatory function.
Yeah. So
but you can mess with the regulatory functions a lot and see very few effects. And in fact, that's one of the main differences between apes and humans. So I think you just asked about what do we see that's conserved between them? We see
over 50%.
We see certain genes only conserved after a certain time period. Especially in the fossil record, we can't test fossils for genes, but we know that genes encode for phenotypes. So for the molecular data that we do have, we see genes conserved among what the phylogeny predicted were close species. And we also use the molecular evidence to make the phylogeny more accurate.
See, here's the problem that I have with these phylogenies and these phylogenetic systematics is that I'm not saying all of it, but a lot of it is based on a primary assumption for one, that universal common ancestry is true. So any type of evidence that contradicts it, they'll just come up with a rescue device, say convergent evolution, for example. But it's also based on an assumption that most of our nuclear DNA differences or evolutionary leftovers or junk or genomic fossils. But now,
are you aware of the phylogeny challenge from Aaron Ra?
Exactly. And the thing is, Aaron Ra assumes that these genetic markers he's using to build the phylogenetic trees are based on evolutionary leftovers. But what I said about junk DNA,
that is the best explanation.
Yeah, but the thing is, what we know about the fact that junk DNA has now been overturned and that most of our genome's functional.
That was more of like a media label. Scientists said we don't know what it does.
But I'm saying now that because you just said that this non-coding DNA is heavily involved in regulatory use, right? For the audience sake, that just means when and how fast to say make specific proteins, but also in embryonic development.
So that's where most of the mutations would occur, and that's just up and down regulation, and that can easily be regulated by natural selection.
Well, are you familiar with, so if we look at like the phylogeny challenge or phylogenetic systematics, a lot of it, like there's specific genetic markers, ERVs, right, endogenous retroviruses. They'll use these, you can say shared mistakes. They'll use these to build the phylogenies. But now that most of this genome is now known to perform many valuable functions, that's consistent with a common design argument now, because now that's proof that they're not actually evolutionary leftovers. Because some of those pseudogenes that Aaron Ra uses to build his trees, they're actually now known, and you probably agree with this, they're necessary and required to sustain healthy life processes in the cell. So how do you explain all of the incoming data on genomic functionality? I mean, that contradicts these phylogenetic trees. How do you explain those?
So one reason that fits best is the likelihood of these mutations occurring the exact same in two different organisms is astronomically lower than you even think that the beneficial mutations are going to be.
Right, I agree.
And so we see organisms that have these shared heritages, and they're conserved. They're conserved among, maybe a type of fish, but not other fish. So why did some fish get it and some fish don't? Only these fish that all kind of look alike and share a common ancestry have the same thing. So along the lines of like the phylogeny challenge, is we would like you to point out at what point the dinosaur is no longer a dinosaur and it's just a bird, and at what point a limbless lizard is a snake.
See,
Or
with the
at what
phylogeny,
point the
I
first
understand.
snakes were snakes.
Because with the phylogeny challenge, what I'm saying in regards to, because we looked at created kinds. So let me just explain briefly, because what I said is these phylogenetic systematic charts are based on the assumption that a lot of this genome is functionally useless. So these shared pseudogenes, which were largely assumed to be genetic mistakes, but now we see that a lot of them are being overturned and shown to be functional. That's consistent with our model of what's called created heterozygosity hypothesis. And what it is if we have heterozygous ancestors, so it's just a fancy word for pre-existing genetic diversity, because we believe God created based on his command to be fruitful and multiply. He didn't want Adam and Eve to reproduce by cloning. That wouldn't make any sense. So with these pre-existing DNA differences, an almost limitless variety of combinations of chromosomes, genes, and traits are then possible. So we look to, Taylor, recombination gene conversion, and what those processes would do would provide many new varieties of combinations of traits quickly, since the differences are already built in. So that's how we explain speciation. We expect speciation, we predict speciation, but there are limits. Now, here's our testable prediction. If God, because a lot of people say, well, that's an ad hoc explanation. Oh, God created Adam and Eve and the created kinds with pre-existing functional DNA differences. Well, here's the thing. That hypothesis makes testable and falsifiable predictions. So we predict that if this is true, the majority of our DNA differences, say nuclear, mitochondrial DNA differences, will be functional. And now what we know about the genome based on ENCODE, based on the accumulating papers indicating function, this is a confirmation of the model. So now these phylogenetic trees are being torn down because these genetic markers, these pseudogenes, these shared mistakes are now consistent with our model of pre-existing heterozygosity. Does that make sense?
A little bit.
Because
I don't understand why. So you fully admit that there's microevolution going on.
Well, I believe, I think
and
microevolution is a bad term to use, but I mean, the evolutionists are using it. It's common. It's a common term. Marvel used it, but obviously, yeah, because.
So but you don't even need mutation really for speciation.
Yes.
Obviously, it's going to help to have different proteins, but theoretically, how long would it take for you to call something a different species? How, like, at what point is a limbless lizard a snake? Does it join the snake kind? Or at what point is, if I breed bears into a small little hamster thing, at what point is it no longer a bear?
Right, and that's a good question, and based on what you said proves that you're listening, so that's really good, because mutations are only going to provide a small amount of variety. So you're right. We actually look to the pre-existing built-in genetic diversity, or in other words, pre-existing heterozygosity, and recombination and gene conversion, which are not mutation related. So based on this allelic variety, allelic potential, visible distinctiveness in offspring is going to be rapid. Okay, so we acknowledge that organisms often change over time, right? Microevolution, variations within a kind, but these are limited to the created kind. So I'm getting to your answer, because speciation, say these similar organisms are isolated from one another, and these DNA differences accumulate until these populations, say they become incapable of interbreeding, for example, speciation has then occurred. So we've made a testable prediction, not only on functional DNA differences, which has been confirmed, but also on speciation rates. So there's a new paper out just recently in regards to finches. And there has been observed finch populations that in observed time have gone from shifts in heterozygosity, so heterozygous DNA sites, to homozygous DNA sites, which we predict, of course, based on pre-existing heterozygosity, and new species, new distinct species of these finches have been observed. So there's a couple papers on that. And that's another confirmation of our model. So in regards to your question on created kinds, we would look to, so we say family level is so whatever I choose, if I choose the species level, the genus level, it needs, testable predictions need to flow from it. That's the gold standard of the science. So we look to the family level. Now we're going to look to functionally unique DNA differences across certain families, because obviously they don't share ancestry. So that's the only way that we can fully answer the phylogeny challenge. We have some answers, say orphan genes, which are taxonomically restricted. But the more we learn about the DNA language, because we only understand less than 5% of the DNA language, we're in the infancy of understanding the DNA language. So the more we learn about it, the more that our model is confirmed, and the more that the evolutionary model is actually being falsified for many reasons. Go ahead.
So do you accept that birds are dinosaurs?
No, I wouldn't, because like I said,
tell me which line along dinosaur to bird evolution, which one was the first bird?
See, the thing is, for me to answer that question, I'd have to assume that universal common ancestry is true. But based on everything we talked about, it's not.
You just have to be able to draw a hard and fast line between them, and you can't, because the gradual change is so slow that no matter which direction you go, you will see gradual change, which you can't draw a hard and fast line on.
No, because
and that's the phylogeny challenge.
But the thing is, the phylogeny challenge is fallacious. For one, it assumes universal common ancestry is true. So it's kind of more so a challenge for R and Raw. But the thing is, our model, our hypothesis that makes testable predictions created heterozygosity, we're now seeing functionally unique DNA differences separating families. So that's how we can understand, and also based on inbreeding and we can understand what's related and what's not. And we can actually conclude that the monkey kind, or say the ape kind, is different than the human kind. So, I mean, there's your two distinct created kinds if you really want.
So what is your criteria for that barrier?
The criteria for the barrier would be,
when is a long-necked horse just a giraffe?
See, the thing is, a lot of what you're saying, say a giraffe and a horse, we have to look at DNA differences, because obviously, you know this being a cell biologist, these phylogenetic trees and charts, they're built by sequencing genes, say mitochondrial genes, for example. So like I said, we're looking at, because I'm not sure if you've ever watched the Dr. Herman Mays versus Ken Hovind debate. Herman Mays asked, what type of testable predictions can you make based on neutral variation? And that's because, based on the assumption of universal common ancestry, those DNA differences, since they share relationships, they're going to be functionally neutral. But we predict that they're going to be functionally unique, because obviously they're not related. So I'm trying to tell you, I might not have every answer to what is a created kind, what is shared ancestry, but what we now know about the DNA is slowly confirming that, because we're looking to functionally unique DNA differences. You guys are looking to functionally neutral DNA differences. But I do want to say this. It's been exactly an hour. It's been longer than an hour. I'm going to let you have the final words here. Sum everything up on what you think. I think it's been a good discussion. And then I got something to do. I'm going to leave it as a free-for-all if you want, just to kind of talk about random stuff. I think there's a couple guys in the chat, and kind of go from there. So I'll leave it to you to say whatever you got to say, brother. Go ahead.
All right. Well, you say that they are distinct, but we can actually make connections between these different kinds. There are transitional forms. And this is just part of the problem, is we understand that there is no hard and fast line. There is no criteria. There's not a set of DNA that says, "Welp, I'm a different kind now." I don't understand where these barriers are coming from. So like, when is a long-necked horse just a giraffe? Giraffe. Speaking of giraffes, I think you've probably heard of this example, but the vagus nerve is a pretty convincing evidence that creation science is not consistent with what we see in reality, because it's a nerve that goes all the way down its neck and back up its neck, which would have no function It's just that way because it's related to other things that have a shorter neck, and the nerve got tangled up basically, and so it keeps its same position while the neck elongates throughout evolution. And it's just these kind of continuations that have no break between all the kinds that's very convincing on the phylogeny. And I guess I'll just close out by kind of saying you guys admit to all these micro changes, but you just kind of assume, it seems like, this barrier that somehow knows when the kind needs to stop changing or when to redirect it so that it doesn't become something that's too different. Because let's take language, for example. It changes only a slight bit at a time, but you give it enough time and it's going to look completely different. It's going to be unintelligible to the people who spoke it just a few hundred years ago. And the same with biological evolution. You add enough changes, it doesn't matter how small they are. It will be different eventually. So that's kind of the position of evolutionists.
Well, I appreciate your. I'll just say a couple last words there. I appreciate your kind of concluding statement there. Like I said, and we agreed at the beginning, we don't disagree with natural selection, mutations. We don't agree. We don't disagree that evolution in regards to biological evolution means a change in allele frequency or a change in gene pool in populations over generations. It has to do with the limits to the change in the direction of the change. So it was my my position that neo-Darwinism, these mechanisms, they lack significant creative powers in regards to obviously anatomical novelty, novel information, phenotypic complexity, but it all came down to beneficial mutations. It all came down to a net loss versus a net gain. And I think it's clear that beneficials cannot keep up with the influx of deleterious mutations and these near neutrals that Taylor and I talked about. I think it's also quite evident that they are going to be virtually unstoppable. So if natural selection has no way to stop the accumulating damage, nor the ability to obviously build a genome based on that, I think, like I said many times, that type of evolution, the pond scum to people type of evolution, that only happens in the imagination of those that actually want to believe in it. And even in regards to the linkage groups that we talked about, that seems to be a killer and a destroyer for particles to people type evolution, because every linkage block is degenerate. So the transitional fossils he talks about are an appeal to the fossil record. Fossils, they're just circumstantial evidence requiring an actual mechanism to explain that type of large-scale evolutionary change. So what we know about observable genomic data, we can safely conclude that it's false, which means whether it's phylogenetic systematics, any other type of evidence, it's being used and it's based on a false assumption that universal common ancestry is true. But since we know it's not true, it's more so a circular argument. So that would be my my conclusion there. And also I want to thank Taylor for being respectful. It was fun, actually. That hour and 10 minutes really flew by.
Yeah.
I think it would be good for us to even do another one because there's just so much to talk about. I think the gold standard of science is testable predictions. So I think I've provided a couple of hypotheses that make testable predictions. So if that's the gold standard of science, maybe next time we can focus even more on testable predictions. So standing for truth and Taylor's portion of the debate is over. It's been fun. It's been great. We'll do it again.